This presentation is a critical appraisal of the TAK-003 tetravalent dengue vaccine trial. It is a 4.5-year phase 3 randomized, double-blind, placebo-controlled study. The four large figures at the bottom give the structure of the trial at a glance. 20,099 randomized participants, 8 endemic countries, 4.5 years of follow-up after dose 2, and a 2-to-1 allocation of TAK-003 versus placebo. Keep these four anchors in mind because they shape how we interpret validity, follow-up, strength, and real-world relevance. Next, we will move from the title slide to the study's overall take-home message. Looking at the summary blocks on this screen, we start with the verdict right up front. The trial provides strong evidence for reducing overall symptomatic and hospitalized dengue in children and adolescents. The overall risk of bias for virologically confirmed dengue is low, supported by excellent randomization and centralized testing. However, notice the text on the right. This is not a perfectly clean story across all four serotypes. The evidence is noticeably weaker for dengue naive participants facing DNV3 and DNV4. Furthermore, there are some concerns regarding hospitalization endpoints because admission thresholds varied significantly by country. The bottom line, highlighted in red, shows a genuinely useful vaccine signal, though tempered by seronegative uncertainty. To understand how the researchers arrived here, we need to examine the specific clinical question they asked. This table outlines the core PICO elements of the trial. The population focused on healthy children and adolescents aged 4 to 16 years in endemic regions. The intervention was 2 subcutaneous doses of TAK-003 given 3 months apart, compared against a saline placebo. The primary outcomes were objective, specifically virologically confirmed dengue and hospitalized cases. As highlighted in the green box below, the trial successfully answers whether the vaccine reduces confirmed and hospitalized dengue overall in pediatric populations. Yet, the orange box points out the lingering unknown. It does not fully establish if dengue-naive recipients get equal protection against all four serotypes, particularly DNV3 and DNV4. Understanding why this gap matters requires a quick look at the unique challenges of dengue vaccine development. Notice the 4 bold numbers at the top of your screen. Dengue vaccine development is notoriously difficult because there are 4 distinct serotypes requiring balanced protection. We must also consider 2 entirely different safety contexts, specifically whether a patient is seropositive or seronegative before vaccination. Furthermore, it takes years of surveillance to detect waning immunity or late onset harm. Ultimately, public health policy demands a clear benefit to risk ratio in endemic settings. Previous experiences with other dengue vaccines prove that prior exposure fundamentally changes this calculation, making seronegative safety a central question. Our appraisal will focus on whether the randomization protected the comparison, if the outcomes are clinically meaningful, and whether the subgroup claims are actually credible. Let us review the trial structure that attempted to address these complex. This slide breaks down the trial architecture. It was a phase 3 double-blind, placebo-controlled parallel group trial. Looking at the top row, the study spanned 26 research centers across Latin America and Asia, enrolling healthy participants aged 4 to 16. Allocation was randomized at a 2-to-1 ratio and stratified by age and region. For follow-up, researchers used weekly contacts to monitor for febrile illness, followed by strict PCR testing. The analysis relied on a Cox proportional hazards model to track cumulative efficacy over roughly 57 months. The red banner at the bottom summarizes our take. This is a very strong design for evaluating overall vaccine efficacy, but we must exercise restraint when interpreting exploratory subgroups. Now, let us see who actually participated in this extensive study. Looking at the baseline characteristics table on the left, we can see a well-balanced participant pool. The key points on the right highlight that the mean age was 9.6 years and the sex distribution was roughly equal. Geographically, Latin America accounted for about 55.2% of the participants, with Asia making up the remaining 44.8%. Importantly, look at the baseline serostatus at the bottom of the table. About 27.6% of the participants were seronegative at baseline, meaning they had never been exposed to dengue. The researchers carefully excluded individuals with pregnancy, immune impairment, or prior dengue vaccination. This careful selection process sets the stage for high-quality data. Next, we will trace how well These participants were retained over the 4.5 years. This flow diagram illustrates the participant journey from initial screening through the final phase of the trial. Out of over 23,000 screened individuals, 20,099 were randomly assigned. If you follow the pathways down to the safety sets, you will see excellent retention rates for such a lengthy study. The appraisal note at the bottom highlights the crucial metric here. R3 completion was an impressive 91%. Furthermore, the discontinuation rates were virtually identical between the two arms, at 9% for the vaccine group and 9.1% for the placebo group. This balanced attrition is a massive strength as it minimizes the risk of bias from missing data. With the population secured, we should examine exactly what was administered to these two groups. On the left, we have the intervention details. TAK-003 is a live attenuated tetravalent vaccine built on a DNV2 backbone given in two subcutaneous doses of 0.5 milliliters at months 0 and 3. On the right, the placebo was a simple saline injection. This straightforward schedule is highly useful for real-world mass vaccination programs. The placebo design allowed for direct efficacy estimation and strict blinding protected against performance and detection bias. However, as the bottom right section notes, there are still unknowns. We do not yet know how booster doses might alter long-term risks and benefits, or how this will perform in routine public programs outside controlled trial centers. To measure that success, we must evaluate how the researchers track the disease itself. This screen highlights the massive surveillance effort required for this study. The primary clinical signal relied on virologically confirmed dengue, which means catching a fever and immediately confirming it with serotype-specific PCR testing. Notice the bold numbers. The team tracked over 27,000 reported febrile illnesses and successfully tested nearly 98% of them during the pre-booster period. This rigorous weekly surveillance caught 17 confirmed dengue cases and 188 hospitalized cases. As noted at the bottom, this objective testing greatly reduces the chance of missing mild episodes. While hospitalization is a highly meaningful clinical outcome, we must remember that admission criteria vary depending on local medical practices.
Let us explore the statistical methods used to analyze these hard-earned numbers.
Here we outline the statistical framework. The core method calculated vaccine efficacy as 1 minus the hazard ratio, using a Cox model adjusted for age and stratified by region. The primary endpoint was specifically powered to rule out a vaccine efficacy of 25% or less at 1 year. However, the right side lists important limitations. Most of the analyses at the 4.5-year mark are technically exploratory, and multiplicity control was quite limited. Once you stratify by serotype and severe disease, the event counts become very small. Our critical appraisal judgment at the bottom concludes that while the main overall results are highly credible, any fine-grained claims regarding specific serotypes, serostatus, or severity should be treated with caution. Keeping that framework in mind, we can now look at the top line efficacy results. The large red numbers on this slide deliver the most clinically important signals of the trial. The cumulative vaccine efficacy against virologically confirmed dengue was 61.2%. Even more impressive, efficacy against hospitalized dengue reached 84.1%. Crucially, there were 0 deaths judged to be vaccine-related. Looking at the horizontal bar charts below, you can see how this breaks down. While overall protection is strong, the efficacy for seronegative participants drops to 53.5% for any confirmed dengue and 79.3% for hospitalized cases. The interpretation box highlights a key takeaway. The vaccine is much better at preventing hospitalization than it is at preventing all symptomatic disease, suggesting a strong disease-modifying impact. To see exactly where this protection varies, we need to examine the forest plot. This forest plot requires careful attention. The overall and seropositive groups show clear protection, with the data points and confidence intervals sitting comfortably on the right side of the vertical line. However, look closely at the seronegative section in the lower half. While DENV1 and DNV2 show protection, the dot for DNV3 crosses the line into negative territory, indicating no protection. Furthermore, the interval for DNV4 is incredibly wide, represented by the long arrow stretching far to the left. This visualizes the sparse data and high uncertainty for that specific group. The text box at the bottom confirms this mixed picture for dengue naive individuals. Let us translate these visual plots into concrete numbers to better understand the serostatus divide. This table directly compares vaccine efficacy based on prior dengue exposure. For overall confirmed dengue, seropositive individuals saw 64.2% efficacy compared to 53.5% for seronegative individuals. Both represent moderate to high confidence effects. Hospitalization prevention remains clinically strong for both groups. However, the stark contrast appears in the bottom two rows. For DENV3, seropositive efficacy is 52.3%, but it plummets to negative 15.5% for seronegative participants, signaling a complete lack of efficacy.
So,
For DNV4, the seronegative data is simply too sparse for reliable inference, dropping to negative 105.6% with massive confidence intervals. The key distinction is that while the vaccine works best in previously exposed children, its benefits in naive patients are uneven. This serotype specific pattern becomes even clearer when viewed over time. These four cumulative incidence graphs map the serotype story over 57 months. In the DENV1 and DENV2 charts on the top row, you can clearly see the dashed lines for the vaccine groups separating from the solid placebo lines, indicating sustained protection for both seropositive and seronegative participants. But look at the DENV3 chart in the bottom left. The red dashed line for seronegative vaccine recipients tracks almost exactly with the placebo line, confirming no observed efficacy. The DENV4 chart on the bottom right shows almost flat lines across the board due to extremely low natural incidence. As the appraisal box notes, this is not a failure of the trial design itself, but rather a precision problem driven by the unpredictable epidemiology of circulating serotypes. This variability also complicates how we interpret severe disease outcomes. This detailed plot examines hospitalized and severe dengue. The overall efficacy against hospitalization is excellent at 84.1%. However, the data gets very thin when sliced by serotype and serostatus. Notice the rows labeled excluding Sri Lanka. Sri Lanka had unusually high hospitalization rates for mild cases due to local admission protocols. When the researchers excluded that data, it reduced some of the apparent numerical imbalances, but the core issue of sparse data remains. The critical caution box at the bottom right emphasizes that while preventing hospitalization is highly important to patients, the heterogeneity of admission practices across different countries makes it difficult to draw rigid conclusions about true biological severity. Beyond severity, we must also evaluate how well this protection holds up in the long run. Looking at the cumulative incidence curves on the left, the separation between the vaccine and placebo lines persists beautifully all the way to month 57. The numbers on the right are striking. During year 4 alone, efficacy against confirmed dengue was 62.8%, and efficacy against hospitalized dengue was a remarkable 96.4%. This persistence proves that the vaccine offers durable protection well beyond the initial immune response. However, our appraisal notes a few caveats. Year by year, estimates do fluctuate because the event counts get smaller over time. Additionally, the final 18 months overlapped with the COVID-19 pandemic, which drastically lowered overall reported dengue incidence. With durability established, we must rigorously examine the safety profile over this extended period. This table summarizes the serious adverse events over the 4.5-year follow-up. The rates of any serious adverse event were actually slightly lower in the vaccine arm at 5% compared to 5.9% in the placebo group. There were 11 deaths in the vaccine group and 6 in the placebo group, which aligns perfectly with the 2-to-1 randomization ratio, and investigators judged none of them to be vaccine related. The green box confirms that this long-term trial shows absolutely no overall serious safety signal. However, the orange box provides an important caveat. What can be done? Because severe dengue events were so rare in this trial, the data cannot completely exclude the risk of rare vaccine-associated enhanced disease in specific seronegative subgroups. This biological uncertainty leads us to look at the antibody responses. These graphs display the geometric mean titers, measuring neutralizing antibodies over time. The chart on the left shows baseline seropositive individuals, while the right shows baseline seronegative individuals. In both groups, antibody titers remained higher in the TAK03 recipients than in the placebo group through month 51. Notice that the red lines representing DENV2 are particularly high, which makes perfect sense given the vaccine is built on a DNV2 backbone. As the appraisal text explains, this immunogenicity provides strong biological plausibility for the vaccine's effect. However, these antibody levels are not a perfect surrogate marker. They do not fully predict serotype specific clinical protection, which is why actual clinical efficacy remains the decisive metric. Let us synthesize all these observations into a formal risk of bias judgment. This table applies a risk of bias 2 style framework to the trial's outcomes. For the randomization process and deviations from intended interventions, the risk is strictly low, thanks to successful stratification and double blinding. However, as we move down the table, you will see some concerns emerge for the hospitalized and severe outcomes. This is largely due to the varying hospitalization thresholds across different countries and the potential impact of the pandemic on late stage data collection. Furthermore, the selection of reported results raises some concerns due to the heavy reliance on exploratory endpoints for the multiple subgroup analysis. Overall, the main efficacy signal remains robust with a low risk of bias, but the highly specific severity and subgroup claims carry more uncertainty. We can summarize the internal validity by looking at the core strengths. The 4 green words at the top highlight why the overall estimate of this trial is highly credible. It is randomized, blinded, objective, and long. The large multi-country sample size and the rigorous weekly febrile surveillance ensure that the event capture is comprehensive and accurate. Furthermore, assessing serostatus right at baseline was a critical design choice that enabled the essential safety subgroup analysis we just reviewed. As outlined on the right, these methodological choices successfully reduce selection, performance, and detection bias. They give us immense confidence in the broad efficacy signal of the vaccine. However, as strong as these elements are, they cannot magically generate data where events simply did not occur. We must counterbalance these strengths by acknowledging the primary areas of concern. This slide outlines where our interpretation must slow down. The primary concerns revolve around the exploratory nature of the cumulative 4.5-year analyses and the lack of strict multiplicity control. When you slice the data by serotype, serostatus, and severity, you run into the sparse events problem, particularly for DNV4. We also have to account for the heterogeneous hospitalization practices like we saw in Sri Lanka and the disruptive impact of the pandemic period on dengue transmission. Finally, we note the industry sponsorship as Takeda designed and funded the study. The red bar summarizes the impact of these issues. These concerns do not invalidate the main overall efficacy claim, but they significantly weaken the fine-grained, highly specific subgroup claims. Understanding these limitations helps us determine exactly where this vaccine can be implemented. This table evaluates how well the trial findings apply to real-world populations. The evidence strongly supports use in children and adolescents in endemic high-burden areas. This keeps showing up high-burden areas. However, support is weak for children under 6, and the trial does not directly support use in adults or the elderly since they were not enrolled. It absolutely does not support use in pregnant or immunocompromised individuals. Crucially, the evidence is highly uncertain for low or moderate transmission settings due to the seronegative efficacy gaps we discussed. The policy lens box at the bottom reflects current World Health Organization guidance, which recommends the vaccine for ages 6 to 16 in high transmission settings, but advises against programmatic use in lower transmission areas until the seronegative uncertainties are resolved. This policy stance directly ties into broader ethical and equity considerations. Looking at the ethics column, clinical equipoise clearly existed at the trial's outset. Because children are a vulnerable population, the reported consent processes and independent safety monitoring were absolutely vital. On the equity side, the multi-country sites beautifully captured the diversity of dengue ecology. However, because the participants were strictly healthy, comorbid populations are underrepresented, and implementation costs were not evaluated. The orange box highlights the ultimate public health trade off. A vaccine that drastically reduces hospitalization can relieve massive pressure on fragile healthcare systems in high burden areas. Yet, deploying it in low transmission areas might expose more dengue naive recipients to uncertain protection against specific serotypes. To clarify this balance, let us clearly separate what this study actually proves from what it does not. This side by side comparison provides a plain language translation for clinicians. It is entirely reasonable to conclude that TAK-03 reduces overall symptomatic dengue and substantially reduces hospitalizations in pediatric endemic settings. The safety profile over 4.5 years looks highly favorable for this specific group. However, look at the red column. The trial does not establish uniform protection against every serotype in naive participants. It does not completely settle the rare severe disease risk, nor does it prove safety for pregnant women or older adults. Therefore, it does not justify indiscriminate use in low transmission areas. The clinical framing at the bottom is clear. This vaccine is an incredibly useful tool, but it is not a standalone solution for dengue control. We can formalize these takeaways using a standard certainty framework. This table applies a grade style certainty rating to the major clinical claims. We have high to moderate, high certainty that the vaccine reduces overall confirmed dengue, backed by a large blinded trial and objective outcomes. The certainty for reducing overall hospitalized dengue is moderate high, slightly lowered only by varying admission practices. Protection for seropositive individuals across all serotypes, as well as seronegative protection against DENV1 and DENV2, holds moderate certainty. However, the certainty drops to low or very low regarding seronegative protection against DNB3 and DNB4 simply because the data is too sparse and the confidence intervals are too wide. Certainty regarding rare severe safety events is also low for the same reason. To improve these lower certainty ratings, we must identify the specific research gaps that still need closing. To strengthen the current evidence base, we need robust post-licensure effectiveness studies. We need large real-world data from high-burden programs that can separate estimates by age, region, and circulating serotypes using harmonized definitions for severe dengue. On the right, active safety monitoring is essential. Surveillance systems must be capable of tracking serotype-specific breakthrough infections and detecting rare severe events like anaphylaxis. The bottom section outlines the remaining evidence gaps. We still need to determine the value and timing of future booster doses. We also need effectiveness data for adults and comorbid populations, clear performance metrics in low transmission settings, and comprehensive cost effectiveness models integrated with vector control efforts. Addressing these gaps will take time, but we can draw a definitive conclusion based on the data we have today. Looking at these four synthesis blocks, the message is clear. Take this trial seriously. It is a well conducted, massive study with objective virological confirmation and impressive long term follow up. At the same time, do not overread it. The clinical data for dengue naive recipients facing specific serotypes is simply too thin to make sweeping claims. The most defensible use of this vaccine is for children and adolescents in high transmission settings, deployed as part of an integrated control strategy. The core clinical message to communicate to patients is that they should expect a significant reduction in symptomatic and hospitalized dengue, but not sterilizing immunity or equal protection across all serotypes. Overall, this vaccine offers a highly favorable benefit to risk ratio in endemic contexts, provided we respect the remaining seronegative uncertainties.
Finally, let us review the foundational literature that made this analysis possible.
This final screen lists the primary sources that informed our appraisal. The foundation of this analysis is the primary trial publication by Trico and colleagues in the Lancet Global Health, along with its extensive supplementary appendix, which provided crucial details on statistical analysis and febrile surveillance. To evaluate the methodology, we utilized the updated COSOT guidelines and the Cochrane Risk of Bias 2 tool. For the broader public health context, the World Health Organization's updated position papers and pre-qualification documents were essential for understanding how these trial results are currently being translated into global immunization policy. Always integrate rigorous trial data with local epidemiological context to ensure safe, effective, and evidence based patient care.